Hello, good morning. It's beautiful to have you back here. Today we'll be talking about historical milestones in forensic microbiology. Historical milestones. So we're going to look at some of the milestones. How this aspect of microbiology came to be. Now, it's important to understand that the history of forensic microbiology is intertwined with the history of microbiology. So, because of course, forensic microbiology came out as an offshoot from microbiology. So, when we discuss the historical perspectives or just look at history of forensic microbiology, it would also be intertwined with the history of microbiology in general as a science. Now, when we discuss the history of forensic, like I said, it has roots in general microbiology. We start from the 19th century. Although it dates a bit earlier, we could start by discussing the work of Anton van Leeuwenhoek, who was a Dutch scientist. He was the first person who made these crude microscopes. He was just using lenses. He was actually a lens maker, and then he made these lenses, and he used these lenses to view very very tiny objects, and he was able to view plant cells, animal cells. He called them animalcules back then. That was the beginning of Van Hoek was the first person who was able to make us realize that there's life beneath what we can see with our eyes. There's a world that is invisible to the naked eyes. So that is where microbiology could have been said to have started. So, of course, forensic microbiology too would have started along with microbiology. From there, we move to the 19th century, where we now have the work of scientists such as Robert Koch and then Joseph Lister. Now, these men were scientists and they did a lot. They worked on a lot of things. They designed aseptic techniques. Robert Koch was involved in the germ theory. So the germ theory has led to our understanding. It's like the bedrock of our understanding of infectious diseases today. So the 19th century saw a lot of these men do a lot of work in identifying different kinds of microorganisms, especially bacteria, and then linking these bacteria to the causation of a lot of disease. So they began to realize that many of these bacteria were pathogenic in nature. This is the work that was done a lot by Robert Koch and Louis Pasteur. Now, in the earlier 20th century, too, we saw other scientists who came and started doing other different types of work, and other different types of microbial techniques started to come to play. And this is quite important for us to understand and follow. Now, it's also important for us to understand that at the 19th century, as it was going, we now started to make use of culture techniques. Before, scientists were trying to culture microorganisms on gelatin, and gelatin has its limitations. Limitations include that it was melting at very high temperature, making it difficult for them to prepare it and then to even culture organisms at temperature that is a bit higher than normal. Now, one of the men, Walter Hess, who was working in Koch's laboratory, you know, his wife was Fanny Hess. She suggested that scientists make use of agar because she had been using agar at home. Agar is something that is gotten from seaweed. We use it to grow microorganisms today. So she suggested to the scientists, and of course, they took her suggestion and tried it, prepared this agar, and found out that it was very good at culturing microorganisms. So, and part of the good thing about the agar is that at high temperature, it doesn't melt that easily. So this helped, and we started to use agars and started to do culture techniques, culturing microorganisms in petri dish. There was also a man called Petri who was the one that also designed the use of these petri dishes, and that was used along with agar to culture a lot of microorganisms. So many people have been coming and lending their input. Now, after the Second World War ended in 1945, and then after the Second World War, the world went into what could be termed a Cold War. So there was still hostilities between the USA and USSR then, where Russia broke out from. So there was still this conflict, and this conflict was like behind closed doors. So Why this conflict is important to us as scientists is that it also helped to drive science in quite a number of ways. The scientists in these countries were trying to outdo each other by trying to provide, weapons that could be used against the opponents. And many of these weapons, some of them were bioweapons, some were nuclear weapons, but some were bioweapons. So there's history is replete with a lot of stories, showing where different types of bioweapons were designed and they were used in stealth mode. So they were used, secretly against one another. And we had the US had was developing its own, USSR was developing its own. We have examples of um, agents such as anthrax, such as botulinum toxin, which we know comes from Clostridium botulinum. We had agents such as Q fever. And then USSR on its own was making agents with smallpox, with plague, and many other kinds of different pathogens like that. The idea was to use it to fight against the opponent. So these things were seen, and these things were, they may not have been positive, but the truth was that they were helping to push forensic science. They were helping to push bioterrorism and the use of bioweapons. So over things continue to escalate, but it was stealth in Cold War. Now it's important to know that in 1972 there was a treaty. A treaty was signed with about 180 countries, and this treaty, the idea behind this treaty was to ask that all countries stop the production and the use of bioweapons, biological agents that can cause harm and damage. That all countries, about 180 countries, signed this treaty. But of course, the Russians, unfortunately, seemed to be running their own programs in private, and so others too continue to run their own programs in private. So these are some of the milestones. Then in the 1980s came the advent of DNA sequencing. As far back as then, the methods that were used were what we called Sanger's sequencing. So DNA sequencing came into play, and when DNA sequencing came into play, scientists began to realize that they could identify different types of microorganisms, and that these microorganisms could be identified at the molecular level. They could be identified on the basis of the kind of DNA which they were carrying. And so this opened the eyes of many scientists, including forensic microbiologists, to the use of DNA sequencing, to 16S sequencing. All of these were from the 1980s onward. Now it's important to note that part of the historical milestones also was the fact that there was an anthrax attack in 2001 in the USA, where some terrorists, used the spores of the Bacillus anthracis, which is a bacteria, and then put the spores on letters. And these letters were emailed to they were mailed, sorry, not emailed, they were mailed to top government functionaries in the US. The idea was to try to infect them with anthrax. But it was not very successful, because although it was able to penetrate through government bureaucracy, but with time it was discovered, and then better steps have been taken to try to prevent such a thing from happening again. Now, from 2010 onward, we now have what we call the high throughput sequencing, the next generation sequencing, which are sequencing methods that are really modern. They are used to also sequence DNA, and to produce results very, very quickly. And then they can work with very large amount of samples and generate very large amount of data. That's where we currently are today in science. So this is important to note, because what we're just discussing is the milestones of forensic microbiology are long, and like I established at the beginning, we said that it is moving along the same lines with your normal microbiology as a discipline. So forensic microbiology is there. So these are just some of the things to note. Then the last particularly important thing we should note is the concept of the Thanato microbiome. The Thanato microbiome, that's a concept which we must understand, is the concept of the microbiome in dead bodies. If human body dies and begins to decay, there's a microbial community in that dead body, and the microbial community changes in composition. There's a succession, there's a change from one composition to another over a period of time. So this just discusses a bit the the historical perspective of the growth of forensic microbiology. I hope we did understand this, and we would continue. We would still run a video where we'll discuss post-mortem microbiology and other concepts. Thank you for listening. If you've made it this far, remember to like and subscribe and share to our friends so that we build this science community. Do have a wonderful day.